complex and diverse, with each part having its own specific duties to perform in controlling cell maintenance and growth. Within the cell, but exterior to the nucleus, is the cytoplasm. This jelly-like substance provides a viscous environment for all of the intracellular components. Over the last several years, scientists have learned that the cytoplasm is not a simple clear liquid inside the cell, but rather a highly complex solution composed of water, proteins, carbohydrates, and lipids. In fact, the cytoplasm, as all the other organelles in the cell, many chemical reactions critical to normal cell function occur in the cytoplasm. The cytoplasm is in motion, moving material such as nutrients from one part of the cell to another. In large plant cells, this motion, referred to as cytoplasmic streaming, is especially common. Here you see the cytoplasm moving around the cell in the space between the vacuole and the plasma membrane. This movement plays an essential role in the plant's life cycle.
That last part where it shows the plant cell, that's something that we normally get to see when we're doing microscope lab, and I'm sorry you won't get to actually work with that directly. We are this week going to be looking at microscopes, and I'll be showing you some simulations with them, and it's just too bad that we aren't able to get together face-to-face to use the equipment. I hope that when a lot of you are taking classes like, for instance, A&P and microbiology, we'll be back to more normal teaching formats, so you're able to get the full lab experience. As it is, we're having to do these simulated labs, and they're pretty good simulations, but it's not the same as the real thing. Questions for me there on the cytoplasm? For instance, where does it show up in the cell? Where would we normally find it? Is it inside the nucleus?
No.
Outside the nucleus. And what is in it? What does it basically contain?
Lipids, carbohydrates.
Yeah. And water.
Protein.
Water. Remember, your body is mostly water, and your cells are mostly cytoplasm, so that's where a lot of it is. Okay, well, let's try another section then. And this one's hard to pronounce. Endoplasmic reticulum. You will very quickly get in the habit of calling it ER. Everyone else does, so that's okay.
Of nutrients, wastes, and cell products within the cell is vital to its ability to function normally. Such transportation is facilitated by the endoplasmic reticulum, or ER, which forms an interconnected highway system on which molecules can move from one part of the cell to another. In electron micrographs, the ER appears as loose strings running throughout the cytoplasm. These structures are actually delicate membranes of sacs and tunnels that carry out numerous jobs in addition to providing a molecular transport system. There are two different types of ER. One type, called rough endoplasmic reticulum, appears to be covered with tiny spheres. Rough endoplasmic reticulum extends from the nucleus and is indeed covered with small structures called ribosomes, which are responsible for the rough appearance. The rough ER connects to the nucleus and is important in transporting certain substances, such as proteins, into and out of the nucleus. Cells that produce large numbers of proteins have more rough ER than those that produce fewer. In addition to providing a surface area for ribosomes, rough ER serves as a storage area for newly synthesized molecules. Another type, called smooth endoplasmic reticulum, has no ribosomes attached to its surface, and thus appears much smoother than rough ER. Like rough ER, smooth endoplasmic reticulum serves as a storage area, mainly for proteins that will later be exported out of the cell.
So, ER. What are these two kinds?
Rough and smooth.
Yes.
And rough.
What is it that makes the rough ER look rough?
They have ribosomes.
Yes, they're little granular things, and I think of it looking like a sheet of sandpaper, where you've got the paper would be, say, the membrane, and then you have these little granular objects attached to it, similar to the grits in the sandpaper. And the key thing about that is those ribosomes, they are in the business of making proteins. Rough ER makes proteins.
What were the two types again? It kind of cut off.
I know, and I'm sorry. Rough and smooth. Rough ER has the ribosomes that attach to it, and it makes proteins. Smooth ER does not have those, doesn't have the ribosomes, so it's not making proteins. It could store them, but it can't build them.
Thank you.
Okay. So ribosomes themselves, well, we have some things on that, and I kind of wish the video had these sections switched around. But
Ribosomes are the most numerous structures in the cell. They appear as tiny spherical structures, may not be attached to the rough endoplasmic reticulum. Ribosomes, which are composed of RNA and proteins, are the primary site of protein synthesis. Since all living things rely on proteins for cell maintenance and growth, it is easy to understand why ribosomes are so abundant. The location of ribosomes within a cell provides an indication as to their specific functions. Those located on the rough endoplasmic reticulum synthesize proteins that will eventually be used in the cell's plasma membrane or exported out of the cell. Ribosomes that synthesize proteins that will be used inside the cell.
I just paused that for a second. You have ribosomes that are attached to the rough ER, others that float freely in the cytoplasm. That was the distinction they were making there. So if you have ribosomes that are attached to the rough ER, then they will make proteins that become part of the membrane that they're attached to, or be shipped out of the cell. Alternatively, you can have proteins that drip freely in the cytoplasm. Those will be made by ribosomes that are floating freely. And these ribosomes are all the same. They come and go, they move around. One cell may have 5 million of them, and they're just moving from place to place randomly. So, again, what do they do?
Produce proteins.
Yes, they do. And proteins are absolutely the most essential stuff of life. Everybody thinks about DNA, but if I pressed a magic button and made all of your DNA vanish, you would not instantly die. If I did that with a different magic button and made your proteins vanish, you'd be dead, all right, instantaneously. So the proteins are more immediately essential to life. They are the living stuff. DNA tells you how to make the proteins, but the proteins themselves are basically what keep you alive. Ribosomes have the job of building those. Okay, let's see about the Golgi apparatus.
Every eukaryotic cell has a uniquely shaped organelle called the Golgi apparatus, named after Camillo Golgi, the scientist who first described this important cell part. Here you see the structure of the Golgi apparatus, which appears as flattened sacs stacked on top of each other. The Golgi apparatus sorts, processes, packages, and delivers proteins and lipids throughout the cell. The proteins and lipids may remain inside used in the plasma membrane, or be exported outside the cell. Thus, cells that produce high quantities of proteins and lipids have a very extensive Golgi apparatus.
That was inside the cell, or used in the plasma membrane, or actually exported out of the cell. Again, the sound cut out. So what would you say that that Golgi looks like?
Flat stacks on top of each other.
Yes. Looking almost like a stack of pancakes, except the individual stacks are not quite touching each other. There's a little separation between those. And what it's doing is some processing of proteins, because when the ribosomes do the work on building a protein, there's often more to be done. They only do part of the job. Some of that gets finished in the ER or in the Golgi apparatus, and then the Golgi helps to direct these proteins to different places where they're going to be used, putting them into compartments that butt off and go in different directions. So it's sort of a shipping and receiving center. Any questions from either? Okay, let's see about lysosomes.
Lysosomes are membrane-enclosed vesicles that form in the Golgi apparatus. They may contain over 40 different powerful enzymes that enable the cell to digest and destroy large molecules. Intracellular digestion by lysosomes, often executed by engulfing and chemically breaking down unwanted cell survival. For example, lysosomes help white blood cells destroy foreign substances such as viruses and bacteria that invade the body. Lysosomes also recycle older or damaged organelles. By engulfing selected organelles and digesting them with powerful enzymes, lysosomes return the digested components to the cytoplasm, where they can be reused. This process helps the cell to continually renew itself. Some organisms use lysosomes to break down food molecules that have been engulfed by their cells. For example, engulfing small prey or food particles. Once inside the cell, small food vacuoles containing the captured substance fuse with lysosomes, which release their digestive enzymes.
Word cut out there, they were talking about amoebas. Those are these small shape-shifting cells that are able to engulf a prey, which is what's gone on here. In this last image, we have the one amoeba has captured 4 smaller green cells, and it's going to digest them using lysosomes. So they're trapped inside of food vacuoles. Lysosomes will then bind to those food vacuoles, release their digestive enzymes, and break these little guys down. When they were talking about cells using lysosomes to break down old or damaged organelles, that happens a lot with our mitochondria. Does anyone know what mitochondria do?
Is it the powerhouse of the cell?
Yes, and we're about to learn something about that here. Well, you have these organelles in your cells. Individual cells may have hundreds or thousands of mitochondria, as you'll see. And they're generating a lot of the energy, almost all the energy that your cells use, and they don't last very long. The mitochondria burn out after about a week, and then they're recycled, broken down by lysosomes, and their molecules are rearranged and put into new ones. So the cells are able to continually renew themselves, taking these older, worn out parts and building new ones. So that'll happen, say, in neurons in your brain. You have these brain cells that might, if you're lucky, last you 100 years, but the individual mitochondria only last a week. Lysosomes are essential in that renewal process. So, mitochondria.
You may have heard the mitochondrion referred to as the powerhouse of the cell. This important organelle generates energy in the form of ATP to keep the cell alive. Some cells may have one large mitochondrion. More often, however, cells contain hundreds or even thousands of mitochondria. Require more energy. For example, kidney, liver, and muscle cells have many more mitochondria than cells that require less energy. Structurally, Mitochondria vary in size and shape. However, all mitochondria have two membranes, an outer membrane, which is smooth, and an inner membrane, which is arranged in a series of internal folds called cristae. The many folds of cristae provide an enormous surface area where vital chemical reactions occur. This chemical activity in turn provides energy for the cell. Mitochondria are semi-autonomous, that is, they can grow and self-replicate within the cell. It contains its own ribosomes and a small amount of DNA. Interestingly, mitochondria resemble bacteria in size and biochemistry, and some scientists believe that eukaryotic mitochondria evolved from ancient bacteria. Considerable evidence supports both this argument and the hypothesis that plant cell chloroplasts are the descendants of bacteria.
So, what is it again that they're doing?
Producing energy.
Yes. Yes. And the volume again stuttered when they were talking about the cells that do the most, but think in terms of how active a cell is. If you have, say, I don't know, maybe a cell that's in your skin, generally not very active. Or if you have, say, one that's storing fat, those are not too active. But consider something like a heart cell or a brain cell, a kidney cell. Those are extremely active, and they are packed with mitochondria. So one cell may have thousands of those inside. They'll fit because they're about the size of bacteria. And as we mentioned, you can pack a lot of prokaryotes inside of one eukaryotic cell. They have this double membrane arrangement, inner membrane, outer membrane. And one important thing which kind of breezed by there, they contain something else that the nucleus also contains. What was that?
DNA. Ribosomes.
Small amount of DNA, and they also have some ribosomes. So that means they're able to actually make some of their own proteins. So that leads to interesting ideas about where they came from, and the going theory now is that they originated as separate bacteria, and they became symbiotes living inside of early eukaryotic cells. It sounds crazy, but there are many examples now of cells living peacefully inside of other cells. And sometimes it's not peaceful, but a lot of times we have these cooperative relationships where cells live inside of other cells. We think that's where mitochondria came from, an early version of that kind of relationship. And over time, they became permanent residents. Over a lot of time, we're talking hundreds of millions of years, very long time. But this planet is very old, and there's been a lot of time for these things to happen in. So a little bit of your genetic code is contained in mitochondria. And if you watch, say, a forensic themed show, they'll sometimes talk about looking at mitochondrial DNA to try to make an identification if they can't get enough nuclear DNA. So keep an eye out for that if you're watching a crime show and that comes up sometime. Thing with chloroplasts is it's the same deal with them. They also have some of their own DNA and ribosomes and make some of their own proteins. We think, again, chloroplasts, as we're about to see, started out as independent cells. They ended up being incorporated into plant cells and into algae. So we'll see those now.
Unlike animal cells, plant cells depend on light as a source of energy. Solar energy is converted to chemical energy, which is in numerous life-sustaining processes. This extraordinary and complex process of converting light energy into chemical energy is called photosynthesis. It is accomplished by highly specialized organelles called chloroplasts. Chloroplasts are the site of food production for plants, algae, and some protists. Chloroplasts may be brown or yellow. They are most often bright green due to the abundant chlorophyll pigments found in their membrane, especially noticeable in the spring. Many plants' chlorophyll breaks down, and what you see as the fall colors of tree leaves are actually other color pigments that have been masked by the chlorophyll during the spring and summer. If we take a closer look at a chloroplast, we see that it has an outer membrane and an inner membrane surrounding an intricate system of connected compartments. Each compartment is made up of individual thylakoids stacked on top of one another. Thylakoids are the sites of the photochemical reactions of photosynthesis.
Okay, so what do they do?
Food production for plants.
Sorry, what do the chloroplasts do?
They produce food for the plants.
Yes, they produce food in the same way that we think of it. If you're fertilizing a plant, you're giving it plant food. That's not really the kind of food that we think of, but internally, the plant needs to have food similar to what we have. That's why we can use them as food. And so chloroplasts are how they make that. They are making complex organic molecules just out of the water and the air. And that's something that we as animals cannot do. We are dependent on them for food. Anyway, they also then contain DNA and ribosomes. So we think, again, there's evidence there that they used to be independent life forms. More to it than that, but I mean, those are some of the reasons that we think that both mitochondria and chloroplasts used to be separate bacteria. You have to go pretty far back in time for when in the case. Skeleton.
When a builder constructs a house, he relies on fence around which walls, ceiling, and entire rooms are built. Similarly, when an aeronautical engineer designs an aircraft, she first produces an intricate wireframe model around which the airplane will be constructed and held together. Cells, too, have an intricate and highly complex skeletal system. Cells have a cytoskeleton, an interconnected system of protein threads that weave throughout the nucleus and cytoplasm, some even extending outside the cell. The cytoskeleton provides the framework for the cell, giving it shape as well as the ability to carry their movements. In addition, the cytoskeleton helps in moving organelles within the cell. The cell relies on the components of the cytoskeleton for locomotion.
So what is it doing for the cell?
It provides the framework or the shape.
Shape, framework, yes. Something else they talked about?
Moves organelles within the cell.
Yeah, movement. Now, related to that is the next part here, cilia and flagella. Those are allowing the whole cell to move, but they're made out of some of the cytoskeleton. So some of the same stuff that goes into the cytoskeleton inside the cell can be put together in a way that protrudes out of the cell and helps it to move, like, for instance, with a sperm cell. So these two are definitely linked topics. Okay, let's see about the cilia and flagella here. I'm sorry? Okay, cytoskeleton, as they talked about it, was basically inside the cell, but the cilia and flagella stick outside the cell. They help the whole cell to move. We'll see that right now. Sure.
Many eukaryotic cells have highly specialized microtubular structures that extend outside the cell's membrane. These appendages move the cell and move fluids over the cell's surface. The two types of microtubules that perform these essential duties are flagella and cilia. Flagella appear as long whip-like tails on many single-celled organs. They also exist on certain kinds of cells in multicellular animals. Some organisms' flagella rotate in a screw-like motion to propel the organism through the water or some other liquid. Flagella often occur in pairs. Some organisms, however, have a single flagellum. Cilia are much shorter and more numerous than flagella. They appear as short, hair-like extensions of the cell. Unlike the rotating motion of flagella, cilia beat backward and forward to propel the cell, as in this paramecium, or to draw fluids over tissues.
Okay, so what are these doing?
Helping the cells move.
Yes. But there's another thing that's more subtle. You have a lot of cells in your body that have cilia, and yet the cells aren't moving. What happens in that case is, say, in your airways, you have ciliated cells going all down your trachea and into your lungs, and what will happen is that you have a system in your lungs where the lungs secrete mucus, and dust particles and viruses and other things that get down into your lungs get trapped in that mucus. And then there are ciliated cells that are beating at the mucus, and they're steadily sweeping it up and out of your lungs. So the cells are staying put, and what they're doing is using cilia to move the mucus past the cells. So cells could use the cilia and flagella to move themselves, or else stay in place and move fluid past the cell. Either way, it's kind of the same difference. So what are differences between these two, cilia and flagella?
Flagella can be singular.
Yes, generally not very many of them.
The cilia are shorter and move backwards and forward.
Yeah, the cilia are really small. Basically, you might have a lot of these little cilia, or maybe one or a few big flagella. Just think in terms of economy, how much the cell wants to spend on those different things. Can you think of any human cells that have flagella?
Sperm.
Yeah, sperm, exactly. You think the classic diagram with the sperm of the tail, that's it right there. So if you want to know what a flagellum looks like, just think of your sex ed. Now we get these ciliated cells in our airways, as I mentioned. Then women don't have the flagellated cells, but they do have ciliated cells lining their fallopian tubes. So those help to move the sperm along. So it's kind of an equal opportunity thing where both sexes end up having some renditions of these cilia or flagella. Well, let's see about the plasma membrane then. Plasma or cell membrane, they mean the same thing.
One of the most important and complex structures of the cell is its outer boundary, known as the cell or plasma membrane. The cell membrane is a thin barrier between the cell's internal parts and its external environment. Since all cells must take in certain substances, such as nutrients, and eliminate other substances, such as waste products, a means must exist to accomplish these tasks and still maintain them between the interior and exterior environments. The cell membrane.
Before they go on, I was waiting for this diagram. I want to point it out to you. You see all these globular structures that are in here?
Mm-hmm.
Yeah, those are mitochondria. So this is a very busy cell. I think this is a liver cell in the diagram. And you can see it's jam-packed with mitochondria. This isn't even all of them. All we're seeing here is a very thin slice through a bigger three-dimensional object. So for this view, we're not even seeing all the mitochondria. But even here, we have hundreds of them in view. At the middle here is the nucleus. You can kind of see the plasma membrane around the outside there. But that gives you, I think, a sense of just how many mitochondria show up inside of cells. And this cell is going to be extra heavy on its mitochondria. This one's very metabolically active and needs a lot of energy. But on average, in your body, about 10% of your body weight is mitochondria. So they're a big part of us. So I wanted to point that out. And also up here, notice how this one is elongated. That's a mitochondrion that's dividing, bacterial style. This one is splitting to make two. So that's replacing one that burned out and was recycled earlier. All right, I'll show you the film again.
Membrane serves two main functions. It protects the internal parts of the cell, and it regulates which substances enter and exit. The cell membrane is selectively permeable, restricting the flow of traffic into and out of the cell to specific molecules. It, in essence, acts as a gatekeeper. The membrane has evolved into a highly complex structure, with each part having a specific function. The cell membrane consists of phospholipid molecules. A closer look at a phospholipid molecule reveals that it is composed of a hydrophilic or water-seeking head and a hydrophobic or water-repelling tail. Interactions between the phospholipid molecules of the cell membrane and the exterior aqueous surroundings force the molecules to cluster together, with the hydrophobic tails sandwiched between the hydrophilic heads. For the cell membrane to function properly, the phospholipids must be fluid. In fact, the phospholipid molecules that make up the cell membrane are in constant motion.
Okay, that went by kind of fast, but what we have are these molecules known as phospholipids, and we'll be getting to know these in our chapter 3. So next week we'll talk some about these. You have a phospholipid, and it has a head group, they're showing here in orange, that's attracted to water. Now, your body is mostly water, cytoplasm is mostly water, your cells have these big watery spaces inside of them, and these phospholipids that are in a membrane will orient so that these water attracting heads will face toward the water. They want to be touching water, but you have these two, they said one, but two water repelling tails. These tails are more like fat or oil. That's essentially what they are. If you think about how oil wants to separate from water, that's essentially what these tails do. So you end up getting a membrane where one surface of it is covered with these water attracting heads, and they're facing toward the watery cytoplasm. Then you've got a region that's avoiding water, and stacked up with that is a whole nother layer of these phospholipids again, but it's facing the opposite way. So you've got more of the water repelling tails and then more of the water attracting heads, and they're facing into a different watery compartment. This is how we can have a membrane separating two different parts of the cell and have different chemistries going on in those. This might be the inside of a mitochondrion, and the other surface here might be facing out into the cytoplasm outside the mitochondrion. We'll be seeing a lot of how this kind of thing works. Don't have to worry about the details of it yet, but basically this is showing the fundamental structure of a cell membrane. It's made up out of two layers of these phospholipid molecules, and the molecules have kind of a split personality, where one end is attracted to water and one end is repelled by water. That's about all I would want you to know about this at this time. We will get into this more later in the next couple of weeks. Are you okay with that at this time?
Yeah.
Sounds good.
Okay.
Fluid characteristic of the cell membrane has been studied for many years, and scientists now refer to the cell membrane as a fluid mosaic model, with a mosaic of proteins found throughout the membrane. The proteins that help make up the cell membrane perform several important tasks, including transporting molecules into and out of the cell. Some proteins are embedded in the membrane and span its entire width. Most move freely and possess channels that permit certain molecules to pass through. Other proteins act as small pumps, actively transporting molecules from one side of the membrane to the other. It's attached to some of the proteins, play a crucial role in helping cells recognize and communicate with each other. This cell...
The part that cut out, they're talking about carbohydrates. These are attached to some of the membrane proteins, and they're part of your immune response, the sort of thing that helps to identify your cells as belonging in you. And if something else comes into your body that doesn't have these right carbohydrates, maybe it has different ones, your body may recognize that as being a foreign invader and try to destroy it. That'll happen with, say, a rejection of a transplanted organ if they don't get that right.
To cell communication is an important line of immune defense in vertebrate animals, since it means that foreign cells can be singled out and rejected.
Okay, and I usually don't bother showing this conclusion. Let's go again and look at the questions, and I'm going to show you where I then have a solution key to this. Well, actually, let's work through this first. So what were some things that the plasma membrane does?
It protects the internal part of the cell.
Yes.
Acts as a gatekeeper for substances.
Right. So it's very much like the nuclear envelope or nuclear membrane, pretty much the same roles. But this plasma membrane is on the outside, and it's marking the outer boundary of the living cell. There may be other things outside, like the cell wall, but those are non-living. This is where we get dynamic, vibrant behavior that we think of as being alive, and that's usually limited to where you get to the plasma membrane. Well, I've got this summary here, and we can talk through this. There are several things on here that apply to everything: animal, plant, bacteria. Do any of these look like they should be in everybody?
The DNA.
Ribosomes.
DNA, yes. Ribosomes, yes. Any others?
Plasma membrane.
Yes.
Proteins.
Proteins, yes.
Cytoplasm.
Cytoplasm. Anything else? I couldn't hear that.
Nucleus.
That would not be right, because bacteria don't have those. Instead, bacteria have this thing we call the nucleoid, which they didn't really go into. But bacteria would get the nucleoid, plant and animal would both get the nucleus. Either way, you're talking about a way to organize the DNA in the cell. How about the cell wall? Do animals have those?
No.
So who does?
Bacteria and plant cells.
You're right about bacteria and plants too. I didn't know if anyone would get that with the bacteria, because we didn't talk much about bacteria here, but a lot of cells do have a cell wall. It's a very popular way to live. So fungi have those, algae have them, bacteria do, plants do. Animals are kind of the exception in not having them. Is there anything that maybe getting rid of the cell wall allows an animal cell to do instead?
Does it allow it for more communication between cells?
More like more movement.
Okay.
Think about if you surrounded yourself in this really thick layer of cardboard or wood. Imagine you're wearing just a suit made up of a big layer of wood around you. Pretty hard to move. Animals have made a different choice in their lives, and they've gone for mobility, which means they're more vulnerable to sometimes injury. If you imagine walking up, say, with an axe to a tree, you swing the axe at that tree one time, it's probably not going down. You swing that one time at a person, he's probably going down. So the plants are more resistant to mechanical damage, but then they have trouble moving anywhere. Animals would instead try to run away from the axe. So the cell walls are very common, and bacteria and plants both have those. We do not. How about the chloroplast?
Plant cells?
Plants, yes. What about bacteria? Do they have them?
No.
No. There are photosynthetic bacteria, but they don't have their own little tiny chloroplasts to do it. And it's kind of the same idea with mitochondria. Who has mitochondria?
Animal cells?
Animals?
And plants. They also do too. Bacteria do not. Even though we think that some bacteria evolved into mitochondria, that doesn't mean that living bacteria have their own little tiny versions of mitochondria. The nucleoid, where was that again?
Bacteria?
Bacteria. Nucleus is in the eukaryotic cells. Okay, plasma membrane, everyone's got that. Vacuole,
Plant cells?
Animal?
that was going to be plant only.
Okay.
Okay. Then the ER and the Golgi apparatus,
Animals?
Animal cells?
eukaryotic cells in general. Those are part of the general complexity that goes in with eukaryotic cells. Otherwise, DNA, cytoplasm, and proteins, everyone has those. So that gets us through generally the scope of what's in cells. What I'm going to do here is go to one section here in, and this is in 6017 right now. I'm going to make the solutions visible there, and then I'll do the same in the other class. Oops.
Where I can see the video again.
Sorry?
Where I can see the video again, like or
All I have with it is the DVD. This is not something that's online.
oh, okay.
This was basically it. I'm sorry, but I wasn't looking this up through YouTube. Okay, so what I've just done is I just made visible for both sections where I have the notes with solutions. So I was giving this question. Now let's take a look at where you can actually see the solutions to those questions right here. So there I've got my answers in italics. So this may be something useful for you to go through and study when you're learning this chapter, chapter 4. So again, questions for me before we do some more? Okay, what I'm going to do then is go back to some of our chapter 1 stuff, spend some time with that for the rest of this morning, and then when it comes to this chapter 4 material, part of it I'm holding until 2 weeks from now. That's the latter part of the chapter 4. Otherwise, on Wednesday, I will be giving you a short piece of lecture where I cover some things that the video didn't. I'll mostly look at some things that you should know, but that were not, I think, covered well enough by the video. So I hope the video and me talking about it together will give you good coverage of cell anatomy, cell structures. But before that, we should look some more at our chapter 1, Basic Introduction to Biology and to Science. So let us do some of that. So we have the scientific method, and that is basically how we have learned most of the things that we know about the world, how we've been able to actually test them and prove many things wrong and show that a lot of other ideas are probably right. This is basically our very well developed way of testing for natural explanations for things that happen in nature. So this could include everything from, for instance, how a bacteria moves to how maybe a hormone works in the human brain to cause a certain kind of mood. But you're talking about natural explanations. We're not talking about whether the garden fairies, decided they liked your garden and that's why your plants grow better. These have to be testable ideas, and that's what happens with natural explanations, is that we can figure out ways to put those to the test. So science is really based around testing, and specifically what we're doing with testing is we're trying to prove things wrong. We come along with a new idea and we go and try to challenge it. Instead of just saying, oh, well, that sounds good, we'll accept that idea, we go and develop lots of tests, and scientists are really in the adversarial mindset, kind of like lawyers fighting in a courtroom. Scientists do that in their own way, fighting over ideas and testing them. As a result, when we come to conclusions in science, we try to keep in mind that we don't know everything yet, and we may still learn things in the future that could change our thinking. So we try to keep an open mind. It's sometimes hard to do, but generally we have the attitude that various ideas aren't proven yet, but they're very strongly supported, and yet they might change over time. Now and then we do get major changes, not very often. It's kind of rare when that happens, but once in a while there is a big upheaval in scientific thinking. And people will sometimes think that, well, that just shows that scientists don't really know what's what, because maybe they're saying cholesterol is good for you this year, and the next year they say it's bad for you, something like that. The point is that if we reject an old idea and bring in a new one, we're getting closer to the truth, even if it means having to get rid of old ideas that we might have clung to for a long time. And that is a strong point of science, this ability to go and make corrections. Now, we have two terms that come up which are often confused with each other. You get hypothesis and theory. And hypothesis is where a scientist has put out some sort of a proposed explanation for something, saying, I think that this is why this thing happens. Hypothesis is in great need of being tested. The theory is sort of a grown-up hypothesis. Theory is something that has been tested numerous times from many different angles, and over time has developed a lot of stature within science. So that's the thing that people get confused when they'll say that something is only a theory, or if they say, regarding maybe why, say, two celebrities broke up, and they say, well, I got a theory about that. That's what people generally think theory means. Theory in science means something different and bigger and better than the way most people consider it. A theory is a grown-up hypothesis that has survived numerous attempts to prove it wrong and has a lot of support from experiment. And here are things that theories will do. They generally have a big scope, and they explain the things that we know. So the things that we know are not just millions of separate little disembodied facts. They give you a framework for understanding what you know, and they make predictions of things we haven't even seen yet. Good theories can do both of those things. So theories are supported by lots of evidence, and they explain the things we know, and they predict sometimes the things that we haven't even seen yet. Here's one. This is the theory that put the sun at the center of our solar system. How did we used to think the cosmos was oriented? What did we used to think was at the center of the universe?
The Earth.
The Earth. Would that work as a roadmap for sending a probe, say, to Jupiter? If NASA still had that model and wanted to send a Jupiter probe, you think it would arrive at Jupiter?
Nope.
Probably would miss the target. Instead, we corrected that with a better map. Initially, it was controversial. I mean, people died over this idea. It was something that was very upsetting to a lot of people who were used to having the Earth at the center of everything. but eventually we learned better, and we end up with the sun-centered model, which then explains the facts a lot better, and it predicts things like if a new comet comes into our solar system, we know that it's orbiting the sun, not the Earth, and the sun has its gravitational influence on it, and that tells us where the comet is going to go next. As a result, the theory lets us predict things we haven't seen yet, like a new comet and where it's going to head next. Another important theory is germ theory of disease. I'm old enough to remember when AIDS was a new thing, when we were seeing HIV/AIDS infection, and it was really scary. That was something where you had generally young men developing fatal breakdown of their immune system, and they would develop something that we called AIDS, acquired immune deficiency syndrome, and they were as good as dead at that point. We had no cure for it, and if they developed the symptoms of full-blown AIDS, and we're talking like 1980 here, it was 100% deadly. So at that time, 1980, we didn't know what was causing it. There were a lot of proposed ideas, but one of those was coming from the germ theory of disease, which we already had a lot of experience with. And some scientists were saying, hey, maybe it's a virus. We should look at that. 6 years later, they found it was a virus. And then they were able to work on antiviral drugs. And today we can suppress HIV infection and prevent people from developing AIDS. So AIDS now is more of a long-term survivable condition, more similar to diabetes. Used to be that type 1 diabetes was 100% deadly before we could do insulin injections. Well, as I said, used to be that infection with HIV was 100% deadly, but it doesn't have to be anymore. Part of the reason it's not is because we already had a good working germ theory of disease that helped us to find a solution to that problem. So I'm bringing these up just to point out that the theory of evolution is at the same rank in science. When you hear theory of this or that in science, these are ideas that are extremely well established, that have a lot of experimental weight behind them, and are not likely to change very much at this point, even though that will occasionally happen. So a hypothesis is what people often have in mind when they're thinking theory. They use the word hypothesis when, or they use the word theory when they should probably be saying hypothesis instead. It's just a common misunderstanding. Questions for me on this? Okay, well, let's see something else here. Let's imagine doing some science right now. Imagine this. Let's say you're going out, you're trying to drive to work, your car doesn't start. Well, imagine your dad's not in the area, your boyfriend's not there to go and fix the car. Maybe you can't afford a mechanic, you'd like to try to get it working yourself. What are some possible explanations for the car not starting?
The battery.
Yeah, so the battery might be dead, sure.
Gas. Water.
Gas.
Out of gas.
Mm-hmm.
Any other ideas?
Mechanical issue.
Yeah, some unspecified mechanical issue. Maybe something really crazy. Maybe someone stole your engine. That would be it. I'm thinking outside the box here. We just came up with several ideas. The thing is, they're all testable. These are all things that are natural explanations, which you could do a scientific test, and that's basically what an auto mechanic would do. He'd do his own version of science to get to the bottom of the problem. And so, when you're coming into science, generally, you have an observation of something. It's interesting. You're certainly interested in why your car doesn't start, and you don't have an explanation. So, hypothesize. One or more hypotheses. Right there, I think we came up with 4 of them. Each of those are ideas that are testable in one way or another. So, what you do if you have one hypothesis is that you can think of what should happen if that's true. That's the prediction. And you can see whether your predictions are correct by doing tests. So if you're saying that, well, the battery's dead, or maybe it's because the battery's dead, you can test that. What's a way to test it?
You charge it.
Yeah, you might try a jump start. Or there are other ways to tell if the battery's dead in the first place. If it's a dead battery, you can make a prediction as to what would happen then. So even before you try jumping it, how would you tell if the battery's dead?
Battery lights.
Yeah, see if the lights come on. Well, you could see if you can get any of the accessories like the radio to turn on. If it's just stone cold dead, that's probably a pretty good place to start approaching the problem. Then you'd go and do the next part, like trying to jump start. On the other hand, if maybe you appear to have plenty of power, but the engine isn't starting, there might be something else. Maybe your starter's broken, for instance, and then you'll hear the starter going whir, 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 nothing happens. Maybe you're out of gas. Maybe someone siphoned the gas out of your car during the night. Each of these things has a prediction that can follow from it, and you can test and see whether those predictions have actually occurred, and evaluate the results. This is showing just one hypothesis and testing it, but really you might have several. Like there, in 30 seconds, we came up with several ideas. But what you can do is evaluate each hypothesis. So, something like this. You get this idealized flowchart model. In reality, it's usually more complicated, but this is the kind of thing that will happen. The observations, when we're talking about science, these are observations made by a scientist who really knows a lot about his field, and he's really up to date on what we know and what we or think we know and what we don't know yet. And he comes up with a hypothesis, and you can think of it as an educated guess, but that's by a very educated person who knows a whole lot about the subject and is able to come up with really good hypotheses. And makes a prediction, tests it, and you either get support for the idea, which means you can do more investigation on that same line, or else reject that and try a different hypothesis. This is a more complex version that's given in your book. So I realize you haven't seen the textbook yet. They've got a more complex version of this, though, and I'm showing it to you now. Imagine you've got an observation. Question. This is suggesting that you come up with 5 different hypotheses. That's kind of like we just did with the car example. Predictions follow from each of those. You do experiments, and those experiments reject some of the hypotheses. Some of the others may still be okay, potentially. You do more experiments, and you're able to get rid of more of those, and you're left with one hypothesis, the last one standing, most likely to be true. So then you investigate that more with further experiments. And all of this is helping you to narrow down a lot of different ideas down to a closer focus on an idea that's probably leading you toward the reality. I like to look at it in this cyclical manner because, in a way, it is a cycle. You go through the testing, you come up with the results, and that almost always generates more observations and more questions. You look at the back of a scientific paper, they almost always finish up saying, we need to do more research on this part and this part. And partly that is an appeal to get more funding, because funding is what powers this. In the real world, it needs money. Typically, a professor may be heading up a project at a university, and he has graduate students that are doing work, and the professor or the graduate students apply for money from places like the National Institutes of Health or the National Science Foundation. They end up getting, if they succeed, they end up getting a research grant, and it describes what they're going to do with the money and what sort of important results they expect to get from the study. And if they got the grant, they do the work. It probably takes about 3 years. They publish results, and that gives them a track record to go back and apply for more money. And this is the main thing that actually university professors are doing. If you're at the university and you're taking classes with professors there, this is what they're spending most of their time on. They have to. The university requires it. Professors may love teaching, they may love being working with undergraduates, but they don't often have a lot of time for it because this is how they're rated. They're rated according to basically how many papers they publish and how much money they bring in, and that's just an aspect of how things work in the real world at universities. You are lucky in a way here at the two-year school, community college, because I'm not doing this. I'm able to dedicate my time basically to you, and I'm not chasing after the money. If this class if you were taking this class, say at the University of Washington, you probably would never see a professor, and it would be taught by graduate students, and yet professors would love to be teaching it, and they just don't have time. This stuff is already taking up 50 or 60 hours a week for them, and they just don't have the time left over. So there's some of the real world experience. Let's think in terms of doing an experiment. So what are we looking at here?
Brain?
Yeah. Looks like someone had a pretty bad day here. I would say so. Fatal stroke kind of indicates it, right? So here's where someone died from probably a ruptured blood vessel in one of the hemispheres of the brain. I'm not sure if that's left or right looking at it. Well, suppose this. Suppose you want to test a drug for lowering blood pressure, because that's one of the leading risk factors for stroke, to have, say, a cerebral hemorrhage. High blood pressure is one of the things that will cause that, because you have these great big blood vessels running through the brain, and if the blood pressure is excessive, sometimes they pop. There are other ways to get strokes, but this one was one of the bleeding ones, and the person probably just had a tremendous headache and went, oh God, and clutched his head and then fell over dead. Probably it was that quick. Well, one of the things that we would look at to try to reduce the risk of stroke is reducing blood pressure, because we have a serious problem in this country with people having elevated blood pressure. I'm one of them. I'm at risk of that sort of thing. I'm taking several drugs for blood pressure myself. So here's one way you could do a study. Imagine you have a new drug and you want to test it out. Let's say you've got 10 people that you want to test the new drug on. Let's say you pick middle-aged men with hypertension. I'd be a perfect one for that study. You give it to them, and then imagine their blood pressure goes down. Does that prove that the drug works, that it's an effective drug?
No.
No, why not?
Because you don't have anything else to compare it to, right?
Yeah. And that is something I'm glad to see just about everybody understands. I've given people a science survey that was done nationally, and I've given that sometimes to my students, and people have problems with a lot of the science questions, but that one just about everybody gets right. So just about everybody understands you need a control. Otherwise, you don't know what to compare it to. So what do? You have a treatment group, and then you give a fake drug to a separate group of people, and they're the control group. And the thing is, nobody knows who was getting the real drug and who got the fake one. The people don't, and the scientist working with them doesn't know that either. That becomes what we call a double-blind study, and that's basically how we conduct these things these days. Why would we need to do that? I mean, imagine we just took 10 people and say their blood pressure is running, 150 over 100, and you give them the drug, and their blood pressure drops down to 100 over 70. Why isn't that good enough to say that the drug lowered the blood pressure?
Because there's other factors like their lifestyles, genes, and other things that come into play.
Yeah, and partly it's mind-body connections. People will demonstrably get improved numbers if you give them a pill and it's somebody in a lab coat giving them the pill and he says, here, I think this will make you better. People will get measurably better numbers out of that, even though it was a fake pill that wouldn't have had any actual biological effect. The mind, overrides the body in some ways. So that's where the control group comes in, because what we can do is we can subtract out that mind-body effect by giving this placebo or fake drug to some of the people and the real drug to others, and no one knows which was which. So double-blind because even the scientist working with the people doesn't know it. It would be maybe a doctor, for instance, who has had someone else provide him with the drugs, and he doesn't know which is which. And so the thing is, they want the doctor who's in contact with the people to treat them the same. Otherwise, he might treat some of them differently if he thinks, well, you're going to die anyway, why should I give you a lot of attention? Or he might even have a financial interest in the drug and just cheat. So you make sure that the scientist or the doctor who's in contact with the people doesn't know which is which. That becomes a double-blind study. You compare the results, and you do that through statistics. And this comes in when you're listening maybe to TV ads and they talk about prescription drugs. There's an important word there, and that is significant. When you hear that word significant, it means that there was a demonstrated effect showing that a drug really did have a convincing effect, improving somebody's medical condition. You prove that by doing the statistical analysis. So imagine this. I have here just a fake set of results. I just made these numbers up. But imagine we've got 10 people that we've given the treatment to. That's the drug we're trying to test. And we have 10 control people that we gave the control placebo to. And I've boxed the groups of points, so we get a cloud of points for each one. And notice up the left-hand side, that's showing how much the blood pressure has been reduced. So bigger numbers are better here if you're looking at whether the drug is effective. Which drug appeared to be most effective? The real one or the fake one?
The real one?
Yeah. Was it convincingly effective?
Not that much, it's just two different around.
Yeah, the point is there's a lot of overlap between those two. So there's a lot of experimental noise going on. And what that means is that if we did the same study with 20 other people, that might just get reversed. There's so much overlap that there's no significant difference between these two. So this is the kind of thing you would analyze and show whether or not the drug was actually working. That's part of real science. It's not part of what we're doing in this class. But if you take a class on stats, you would get to do this sort of thing. Here's a different possible set of results. Here I kept the controls the same. Notice this just contracts basically to be this group. And instead, I made the treatment ones much larger. And so now one of these went down by as much as 25 points. Now, does it look like there's a real difference between the treatment and the control groups?
Yes.
Yes. And if you do the statistical analysis, which I did, it indeed shows that the treatment had a significant improvement over the alternative. This is what a medical company would be looking for with their new drug. And it takes millions of dollars developing these new drugs. Ideally, they get these kinds of results instead of this kind. But sometimes it doesn't work, and then you have to just say, well, that's millions of dollars wasted, but at least we know we don't need to waste any more time with that thing. We'll try something else. So these are some of the things that come in with modern science. Let me show you something else, and this is kind of a horror story, but it's instructive because this helps us get to the modern germ theory of disease. You go back into the 1840s, we had lots of cases where in Europe mothers would give birth in a hospital, and they would get sick. They would come in healthy, and they would be in, it would be a medical emergency because they're giving birth, but they're otherwise healthy. But they would end up getting sick with something when they were in the hospital and die. So they would come in, give birth, and die without ever leaving the hospital. It was known as childbed fever, and it killed a lot of the mothers, also sometimes killed the babies. So this was an ongoing, terrible problem that they had in hospitals at that time. And we understand now what was going on, but at that time they didn't. Bacteria were getting into the mother's bloodstreams. Childbirth is a really messy process, and you would sometimes get contamination of the mother's bloodstream with bacteria, and then you'd get a raging septic infection with a bacteria that is called Streptococcus pyogenes. And magnified, the strep bacteria look like this. And you'll, in microbiology, get to look at similar things, little chains of these bacteria. This particular kind causes us problems now. People get strep throat from this stuff, which is a serious infection. Even some of the stuff you hear of as a flesh-eating disease is caused by Streptococcus pyogenes. Well, do you think we had any antibiotics against this stuff in the 1840s?
No.
No, we didn't have any antibiotics. We had no idea that these things were dangerous. All we could do was barely make out these tiny little dots under the best microscopes at the time, and we had no clue as to how dangerous these were. So you didn't really have an immediate way to look at these and say, well, we need to protect against these bacteria. We barely understood that bacteria even existed. So you had this medical mystery. All these women died. This guy, Ignaz Semmelweis, he took us a long way toward figuring out the answer. 1840s, he's managing 2 maternity wards in this hospital in Vienna, Austria. That was the biggest hospital in the world at the time, and people in Vienna were very proud of it. There they had the biggest, most modern hospital anywhere, but they had this horrible rate of infection with the mothers. And it turned out there was a natural experiment already set up involving a treatment and control if you knew what to do with it, because they had 2 wards in there, and these wards were the same, but they used different staffing. One staff, it was modern. They were using med students. The other one, they were doing it the old-fashioned way with midwives. Notice where the infections were showing up. It's medical students. That's where you were getting the high infection rate. Can anyone guess maybe why?
They're not washing their hands.
Somebody already peek and know the answer? Because you're right. So, the difference. Well, here's what the hospital looked like. It was a big facility. Well, again, we didn't understand yet about what bacteria could do. And here's the question. How do you think medical students then were studying human anatomy?
Cadavers?
Yeah, which they do now. It's the same thing. I mean, there's a reason to do that. The thing is, now they're obsessive about sanitation. Back then they weren't, because they didn't know any better. So these medical students would be studying a corpse which was teeming with bacteria. They would also be doing autopsies on women who had just died from childbed fever. And the doctors weren't gloving up, and they weren't sanitizing. They didn't know better. So their hands are just crawling with these bacteria. And then there would be a call for someone to help with their birth, and the guy would run from the autopsy ward right to the maternity ward and help with the birth. Guess what happens? Healthy woman gets infected and she dies. So you had this horrible circle of infection involving the people who are trying their best to save lives, not kill them. That's the horrible tragedy of this. But some clues came up. One was that one of the male doctors got a cut when he was doing an autopsy, and he died a few days later from something that looked just like childbed fever. Well, this guy had not just given birth, he died. Also, beyond that, some of the babies, newborn babies, were dying from this disease that looked like what their mothers were dying from. So we're getting these clues. And Semmelweis comes up with these observations. You've got a lot of women dying from childbed fever. Some of their babies are dying with apparently the same thing. And this male doctor dies from what looks like the same thing. The great insight is that it is the same disease in every case with the same underlying cause. So, Semmelweis thinks somehow it goes back to the bodies, to the cadavers. He thinks that there's something with the cadavers that might be spreading this disease. He's not thinking of bacteria because no one was, but he thought that somehow contacting the cadavers might be the key. Some sort of way that infection is being passed from those bodies into live people, and the doctors are the spreaders of it. So he ordered his doctors to use a chlorine hand wash to sanitize their hands. Chlorine, what does that kind of sound like?
Bleach.
Yeah. Well, bleach is an excellent disinfectant now. So that was a terrific choice if he was going to pick something randomly. Well, he didn't pick it randomly. I always wondered about that. Why bleach? Because that was brilliant, but was it just a wild guess? Now, actually, if you were a medical student, you were going to be living in elevated levels of society. I mean, being a doctor, that's a pretty good job. And so these medical students, in the evenings, weekends, would sometimes be out at society functions with, cultured ladies and so forth. They didn't want to go in there smelling like dead bodies that they'd been handling, so they would clean up using this chlorine hand wash. They smell like bleach, but that's better than smelling like a dead body, right? And Semmelweis thought that if it kills the smell, maybe it kills the cadaver matter too. And he was right, because it did. So these doctors, he orders them to always wash up with this hand wash before they come to visit one of the women. And that cuts the infection rate down to the same as in the midwife ward. So here's where you had in the ward 1, this is the one where they kept getting the high infection rates. Going through the 1840s, about 6 years worth here, rates sometimes dropping low, but going back way high, like in this case, 32% of the women coming in dying from this disease. At other times, much lower. The rate is hopping all around here. Once they went in with the chlorine hand wash, the rate dropped low and it stayed low, about the same as what the midwives were getting. So here's evidence that the idea actually worked. This idea of a connection with the dead bodies is established. So this was an early medical intervention involving scientific thinking that saved a lot of lives. So we have the multiple observations, as I mentioned. We have this hypothesis that it gets back to the cadavers in every case. Prediction that the sanitation will stop it, tested it, and it worked. So did this catch on worldwide? Did this become popular everywhere? Do you think this became then the standard for medicine?
Yeah.
Yes.
We'd like to think so, but no, it didn't. At least it did at that hospital, but I have heard that for various reasons it didn't. Partly, Semmelweis was a pretty abrasive guy, and if he was telling people something, they didn't necessarily like the messenger very much. Also, consider the message he's telling them. He's going to other doctors and saying that, well, when you've been trying to save lives, actually you've been killing them. That's not a very popular message. A lot of people didn't want to hear that message. And also, he didn't have anything to actually point at to say what the cadaver matter was. That came later when Louis Pasteur was working with bacteria in the later 1800s, and he demonstrated that it really was bacteria causing many diseases. And that was decades after this happened. So, for various reasons, this took longer to catch on. But this is an important reason for why they're obsessive about washing hands in hospitals. This is part of the learning curve that went into that. Okay, so that is what I am going to stop with right here today. You guys, I'm going to want you going into your canvas and trying to then go to the Connect system and setting up your account with McGraw-Hill. After that, you should be able to then start working and seeing the lab assignments for tomorrow and for Thursday. So that's basically your first assignment, is to go and try to connect with the Connect system. I am otherwise going to be reopening this page for office hours in about 25 minutes. I can take a question right now, but then I'm going to separate from this, and I'll be back here and reopen this at 11:15. And I think that's typically what I'll be doing for office hours.
Are you updating Canvas to show that we did the labs? Because when I did the lab, it didn't show that Canvas it was complete.
I'll have to look at that. Is that Riley speaking?
Yes, speaking.
Okay. I'll take a look at that. It should automatically upload that, so if it didn't, I'll take a look at it and see why it didn't.
Mine did.